Hello everyone. Today's class will be on the topic hemostasis. The class will mainly be about the steps of hemostasis and how the temporary hemostatic plug is formed along with the secondary hemostatic plug. So this topic is a very important essay and it can also come as a short essay or as even a short note, mainly some parts of it. So when we're coming to hemostasis, it can be divided into three different steps, that is vasoconstriction, then temporary hemostatic plug formation, along with that blood coagulation, and finally the secondary hemostatic plug formation. So if we're looking at the mechanism of hemostasis, it all starts with the vascular injury. So the endothelium is always trying to maintain its integrity, and whenever there's a vascular injury that takes place, these three steps will be initiated. So first, obviously, there will be the mechanical constriction. So mechanical vasoconstriction will be followed by chemical vasoconstriction, which will lead to the temporary hemostasis. Along with that, the collagen exposure always activates the platelets and causes platelet adhesion and aggregation, along with the platelet activation and release. That will help in the formation of the temporary hemostatic plug, and that also leads to temporary hemostasis. This chemical vasoconstriction is aided by many different chemical mediators, which we will be getting into further. So then the final process is the formation of the secondary hemostatic plug, that is with the release of the tissue thromboplastin, which activates the clotting and then forms the definitive hemostatic plug, that is this, and then there'll be the permanent hemostasis. So this is the entire the entire hemostatic pathway in a shortened version. So let's get into it in detail. So then first coming to vasoconstriction, which is the first step, it occurs due to the contraction of the vascular smooth muscle in response to the injury, that is due to the injury of the endothelium. So that helps in decreasing the blood loss. As we have already learned, it leads to temporary hemostasis, and it also helps in activating the platelet plug formation. Then we have the contraction of the smooth muscle, which is initially a mechanical response and later a chemical response due to the secretion of vasoconstrictor substances such as serotonin or thromboxane A2, which we will discuss later, from the activated platelets. So vasoconstriction, as we already said, it's initially a mechanical response which is then mediated by chemical mediators. So that's all about vasoconstriction. Now coming to the temporary hemostatic plug formation. So the temporary hemostatic plug formation, it is mainly with the help of three platelet properties that this temporary hemostatic plug is formed. That is with platelet adhesion, aggregation, secretion, also known as release reaction. So the platelet adhesion, then we have the platelet aggregation, and then finally the platelet secretion. So the platelet adhesion, the platelets have a high affinity of adhering to the exposed vessel wall. Exposed vessel wall meaning the wall which has undergone vascular injury. The exposed collagen and the von Willebrand factor which is there in the injured vessel wall attracts the platelets due to the receptors in the platelet cell membrane. So what are these receptors in the platelet cell membrane which will go and attach the von Willebrand factor? It is the glycoprotein 1b9 receptors present on the platelet membrane, which will mainly help in platelet adhesion. So it is also controlled by the following factors. It obviously depends upon the depth and the degree of injury. That is, more deeper the injury, more platelet aggregation due to the quantity of platelet activating factors from the tissue. That's more amount of platelet activating factors are released from the tissue. Then it also depends on the site of the injury. Platelet aggregation is usually more in the mucocutaneous tissue compared to other regions. Then when we come to the age of the individual, platelet aggregation adhesion is mainly less in elder individuals. Then coming to the hematocrit, when we look at the hematocrit, it actually, when there is increased amount of PCV, there will be increased platelet aggregation. This is mainly due to the increased amount of red cell volume that actually causes more amount of RBCs to be present in the middle part of the axial flow, and it will push away the platelets and the leukocytes towards the edges. That will also cause increased platelet aggregation. That's why when there is decreased PCV, like in cases of anemia, we'll see decreased platelet aggregation. Then coming to the speed of blood flow. Obviously, if the blood flow is more sluggish, there is more chance of platelet aggregation. Then the size of the blood vessels. This determines the number of platelets passing through the vessels at a given time that influences the platelet adhesion. So it just depends upon the size and the number of platelets that are passing through the vessel. So that is about the platelet adhesion. Then now coming to the next step, that is your platelet aggregation. So platelet aggregation, that is aided by fibrinogen, glycoprotein 2b3a and thrombin. So when we're looking at this picture, we can see that the fibrinogen along with the glycoprotein 2b3a helps in the aggregation of platelets. So glycoprotein 2b3a is a receptor which is present on the membrane of the platelet, and then obviously fibrinogen helps in interlinking glycoprotein 2b3a receptors present on various different platelets. So now the platelets are activated by chemicals from their granules, for example ADP, platelet activating factor, etc. And thus the accumulation of more platelets at the site of the vascular injury facilitates the formation of the temporary hemostatic plug. So along with this platelet aggregation that was facilitated by fibrinogen, glycoprotein 2b3 and thrombin, we'll obviously need the next step also to happen, that is the platelet activation and the release of the contents from the platelet granules. So adhesion of the platelets to the damaged endothelium activates the platelets, and it leads to the polymerization of the microfibrillar actin which is present in the platelets, and it leads to a pseudopodia or spicule formation. So because of this activation, it leads to the formation of these pseudopodia or spicules that are seen on the top of the platelets. And along with that, what happens is that through the canalicular system, various mediators are released, and that is known as the release reaction. So the mediators released are released due to the activation of two granules, that is you have your alpha granules and you have your dense granules. So the alpha granules, they mainly consist of the other mediators that are needed for more amount of platelet activation and adhesion. So for example your von Willebrand factor, the thrombospondinin also helps later in the platelet pathway for the secondary hemostatic plug formation, and platelet activating factor is also an important factor, and all of these together help mainly to strengthen the temporary hemostatic plug, and they also help in aiding the formation of the secondary or the definitive hemostatic plug. Then you have the dense granules such as which release the serotonin, ADP, calcium, etc. So these factors are mainly known to cause vasoconstriction. They help in aiding the temporary hemostatic plug formation. For example, serotonin is a good vasoconstrictor, and as we talked about thrombospondinin before, it also promotes the exocytosis of the granules and more release of products from the granules. So the mechanisms through which these granules are released is through mainly the platelet activating factor. As we already said, it is a cytokine which is present in the alpha granules, and this cytokine acts via G proteins. It activates phospholipase C and diacylglycerol, which increases the cytoplasmic calcium. So we also know calcium is needed for the coagulation pathway to take place. So obviously that also helps there, and it also causes microfibrillar contraction and helps in the release of granules. So altogether, these factors which are released from the granules help in aiding the formation of the temporary hemostatic plug, and also in the future the definitive hemostatic plug. So till now, through this we saw that there is a vascular injury at the site of the vascular endothelium causing platelet adhesion, which was the first step, and exposed collagen in the damaged endothelium is what activated the platelets to help in the platelet adhesion. After that, we saw that there was platelet aggregation with the help of fibrinogen and glycoprotein 2b3a, and after that we saw there is the formation of the fibrin thread in the platelet plug. So the blood coagulation and the formation of the secondary hemostatic plug. So before that, we need to know about the clotting factor. So there are 13 clotting factors, but factor 6 does not exist. Other than that, all the other factors are there from 1 to 13. So the first factor is known as fibrinogen, then prothrombin, tissue thromboplastin, calcium ion, factor 5 is proaxlerin, factor 6 does not exist, and then factor 7 is proconvertin, factor 8 is anti-hemophilic factor A, which is actually needed because if it is not there, we have a congenital condition known as hemophilia A. So then factor 9 is Christmas factor, anti-hemophilic factor B in its absence, which causes hemophilia B. Then we have the Stewart-Browder factor, which is an important factor. Then plasma thromboplastin antecedent, which is the factor 11, also known as anti-hemophilic factor C, which also leads to hemophilia C. Then you have the Hagman factor, also known as the contact factor, which initiates the intrinsic pathway. in its start we can see the factor 12, also known as the Hageman factor or the contact factor. Then we have the fibrin stabilizing factor, and we have other factors which are not actually directly related to the pathway, but they are needed for the pathway to function. That is your high molecular weight kininogen, precalcrine, calcrine, and platelet phospholipid. So when we come to the blood coagulation pathway, there are three major steps that we follow. The first one is the activation of the Stewart-Browder factor, also known as the factor 10. And it mainly has two pathways, that is your intrinsic pathway and the extrinsic pathway. So first, after this step is done, that is the intrinsic pathway and the extrinsic pathway, which leads to the activation of the Stewart-Browder factor. These two pathways, they coalesce to help in the formation of thrombin from prothrombin. Then finally we have the formation of fibrin from fibrinogen by proteolysis of the soluble fibrinogen, polymerization of the fibrin monomers, and stabilization of the fibrin. So first we'll go into the stages 1 and 2, that is the activation of the factor 10 or the Stewart-Browder factor, then thrombin, and then finally fibrinogen. So first, coming to the intrinsic pathway, the first step is the activation of the factor 12, which is done with the help of the vascular injury and the contact. Because the contact helps in it, this is why it's also known as the contact factor. The exposed collagen, the negative surface charge, and the high molecular weight kininogen and calcrine are what help in the factor 12 in becoming the factor 12A. Then we have the activation of the factor 11, which becomes factor 11A with the help of high molecular weight kininogen along with factor 12A. The factor 11A then helps in the activation of factor 9 to factor 9A, which is also accelerated by calcium along with factor 11A. Then we have the step 4, which is the final step of this stage, which is known as activation of the factor 10A. The activation of the factor 10A is done not only by these factors which are seen here, that the activation of the factor 10A is mainly done with the help of platelet phospholipid, calcium, and the activated factor 8A. How is the factor 8 activated? The factor 8A is activated after it is separated from the von Willebrand factor in the circulation. So the activation of this factor 8 is also facilitated by this factor 10A. So it is a circular process. So hence, with the activation of factor 8, we also get the activation of factor 10, and with the further activation of factor 10 also activates the factor 8. So it is kind of like a recurring process. So this is the final step in the activation of factor 10 or the activation of Stewart-Browder factor. So this is an enzyme cascade hypothesis, because the every activation of the clotting factor helps in the activation of the next clotting factor. As you can see, it's a cascaded process. So it's also known as the enzyme cascade hypothesis. Now coming to the intrinsic pathway. The extrinsic pathway. We're discussing about the intrinsic pathway now. The extrinsic pathway is quite less complicated when you look at the intrinsic pathway, because it only has majorly 3 steps. So because of the vascular injury or tissue damage, extrinsically, it leads to the release of the tissue factor, also known as tissue thromboplastin. This tissue thromboplastin, what it does is it activates the factor 7 to factor 7A and helps in activating factor 10A. So this tissue thromboplastin is the factor 3, and this is the factor 7. So these two together, you can remember this, because factor 3 along with factor 7 helps in activating factor 10. So just the sum of this helps in activating the factor 10. So the factor 7A along with platelet phospholipid, calcium, and tissue thromboplastin helps in activating factor 10A. So the easier way to remember the extrinsic pathway is just 7 plus 3, which equals to 10. Over here, how you can remember this is that we know that there are 13 clotting factors, and this starts with 12. So it goes in the descending order from factor 12A. It then goes to 11A, or we already know 10 is over here, so we have skipped 10. So then after 11A, we skipped 10, and then we came to 9A. And then obviously 9 will help in activating the stored power factor. So that is the stage 1. So that's how you can remember this a little more easily. And now coming to the activation of thrombin from prothrombin. So prothrombin to thrombin. Thrombin is the second tissue, is the second factor, or the factor 2, and it is activated with the help of platelet phospholipid, calcium, and activated factor 5A, also known as proaccelerin. So it is factor 5 is known as proaccelerin because it also accelerates the conversion of prothrombin to thrombin. As we have already learned over here, proaccelerin is factor 5. And this is the factor 7 is also known as proconvertin because it helps in the conversion of 10 to 10A. So what all does thrombin help in doing? Thrombin is one of the major factors which helps in making the fibrin monomers. It helps in the formation of the prothrombin activator. So thrombin also helps in activating prothrombin to form more thrombin. So that's also another one of its function. It is a potent activator of the platelets, and another one of its functions are it also has an anti-clotting activity. It binds to the factor thrombomodulin on the endothelial cells that allows protein C to be activated. It also has a anti-platelet or anti-thrombotic activity by binding to thrombomodulin. Then it also helps in the repair of the vessel wall by the formation of the clot or the thrombus. So that is the stage 2, or the activation of prothrombin to thrombin. Then along with thrombin, calcium also acts to help in converting fibrinogen to fibrin. So fibrinogen to fibrin is formed, and the fibrin will be stabilized with the help of the factor 13, which we have still not seen. So the factor 13 is one of the factors which act at the end, and that also helps in forming the stabilized fibrin polymer or the fibrin clot. So we had told that the formation of fibrin from fibrinogen has 3 steps. That is, as we discussed here, the proteolysis of the soluble fibrinogen, the polymerization of the fibrin monomers, and then finally the stabilization. So the stabilization step is helped by the activated factor 13A, which helps in forming the stabilized fibrin polymer or the stabilized fibrin clot. And then the other mechanisms that also help are obviously thrombin and calcium. So fibrinogen, with the help of thrombin and calcium, is made into soluble fibrin monomer, and there'll be release of the fibrinopeptides. These fibrin monomers will join to form protofibrils that aggregate to form thick fibrin fibers, and these fibrin fibers undergo polymerization and they become insoluble fibrin polymers, and this there'll be cross-linking of these fibrin polymers. So these fibrin polymers will cross-link to each other, and it will help in forming a definitive hemostatic plug, as we have seen in the above picture. This diagram is a very important one that you have to remember, and along with that, this pathway. And if you remember the major 3 stages, then it just becomes easier to understand what are the different clotting factors which are involved. Like for example, in the extrinsic pathway, it's 7 plus 3, and over here it's in the descending order from 12 to 9, excluding 10 because 10 is the stored power factor. So I hope you guys have understood the formation of the secondary hemostatic plug and blood coagulation along with the temporary hemostatic plug. So thank you everyone, and I hope you enjoyed the class.